Amplification of the fragments before we give it to sequencers, that is an important step in next generation sequencing. We do that with the help of PCR. There are two kinds of PCRs which are performed in different technologies. One of them is called as emulsion PCR, in which the PCR reactions are performed in a solution that is mainly made by emulsion oils. And the second technique is where we do those PCRs on the flow cells, so that is called as Polony PCR. Let's look into the workflow one more time. So we extracted our sequences, we prepared our libraries, and then we are amplifying them. So in order to do that, we see that there are two ways. One is emulsion PCR and one is Polony PCR on our slide. Emulsion is mainly done in OinTorrent 454 and solid machines, whereas Polony is a feature of Urbana sequencing. So in emulsion PCR, as we have seen that if we look into the 454 machines, we have adapters in which one of them contains the biotin, so that helps attaching these fragments to a streptavidin coated beads. The ratio of the beads to the DNA molecule is controlled so that each bead must have only one fragment. So we take the bead, we attach one fragment on it, and we amplify it over there. The purpose of this amplification is to get many copies of that sequence, and then obviously when we sequence that, what will happen is that we will have strong signal. So if we have only one kind of fragment in each bead, we will have just multiple copies of that, the same sequence. So, and then our signal will be strong, and that will be a stronger evidence in order to detect a correct nucleotide. Emulsion PCR, as we have discussed earlier, so we added an oil which is an emulsion oil, and then we put those beads which are coated with streptavidin, and then we have our fragments over there. So if we look into the cross section, we observe there are different droplets within that oil, and each droplet contains a bead and individual fragments. So these are kind of our PCR as well as the sequencing stations. We call them as microreactors. Here is just one microreactor in a good magnification. So you would see there are just one single stranded fragment, and then we have the beads in which we have sequences which are attached to those beads, so that are complementary to this what we have over here in this DNA fragment, so it will help its attachment. So each bead ends up coated with about 1 million of identical copies of the original DNA after the PCR is done. And then this is, if we look into in detail, so we get those fragments, and then we have selected one droplet where we have just one fragment, and then this fragment gets amplified over here, and then they are then given into some microtiter plates, so in which we have those wells. So each bead then rests over there where the sequencing reactions they take place. We will look into sequencing later on. Polony PCR, Polony stands for PCR colonies. This is a feature of Urbana sequencing, where what we have seen here is that we have flow cells, we have slides in which we have lanes, which are actually the watertight channels in which this amplification is performed as well as sequencing is performed. So we have actually clusters on those lanes. We have different clusters where this amplification and sequencing is performed. There is a lawn of primers that is attached, that attaches the sequences while they come into those lanes, and they attach them through their adapters. These reactions they occur in the flow cell, which provides large surface area where we can have huge number of sequencing reactions in parallel. Here we again see the dissection of a flow cell. Basically it's a slide-like thing. It has different channels, or what we call them as lanes. So each lane can take different samples. So nowadays there are the lanes that are having 15 to 16 different samples. If we look into the cross section, we see that there are different primers which are attached to the surface of those lanes right there, and the incoming sequences they get attached to these primers through the help of their adapters. So here we see the cluster generation, in which those molecules which need to be amplified, they come inside and then they get attached to the primers which are attached to those flow cells, and then after that those primers are extended. So in the end we get a new strand. So these new strands are actually covalently linked to the flow cell surface right here. So we don't need the old strands, so we just simply discard them. So those single strands, then they flip over to the adjacent primers, and then in this way they form a bridge-like arrangement. We call it as a bridge. So and then those second primers, they get extended, and in this way we get this amplification. We call it as bridge amplification. Then later on that bridge is denatured, and then the two strands are set free. Then those strands and many other strands, they do the same thing. So this bridge amplification is repeated multiple times, and in the end we get something like this. So we get so many single stranded strands whose reverse strands have been cleaved away, and now they are ready to do the sequencing step. So what we have seen so far here is that fragments, DNA fragments, they need to be amplified, and for amplification there are two major techniques used in these next generation sequencing technologies. One of them is emulsion PCR and the other one is Polony PCR.